Okay, hi everybody. I wanted to give you a real quick um, look over as far as mitosis. So I will go through this, and I'm going to start with the G1 phase. So I might annotate a little bit. If I can get there we go, with my drawing tool. Okay, so in the G1 phase of interphase is your growing phase, okay? And the cell is growing and increasing in size. And at the end of it, there's kind of a little stop sign. Oh, this is a horrible drawing of a stop sign. Stop, okay? And it's a stop sign, and there are 3 Ds that happen here. The first one is to divide. You can go on and divide. So you are going to go on and divide as a cell. Okay, that's your first choice. You can differentiate. There are some cells that do not divide and will not divide. And so they would just hang out and be cells. Like, for example, red blood cells don't divide. Okay, so they would differentiate. Differentiate means become specialized, and they don't go on. And the last one is die. Okay, so you can divide, you can differentiate, or you can die. And this is a really cool word, apoptosis. And what happens is the cell actually pops. Do you remember us talking about lysosomes, that they contain digestive enzymes that can cause the cell to go through cell suicide? That's exactly what this is. So between the webbings of your fingers and toes, the cells die. Did you know that? Some people don't have that die, but there are times where cell death is something that has to happen. Okay. So something to, if we're going on, so otherwise you would hang out here. Otherwise you'll go on and divide. So I'm going to erase here that piece. All right. So we're going to go ahead and go on and divide. So if we're going to divide, one of the things we have to do is we need to make sure, if I'm going to divide myself, I want to make sure that I get an identical copy. So I need to multiply all of the things inside of me. So as far as the DNA, you need to go through DNA replication, where replication. So for example, if you have 46 chromosomes, you would double them to 92. Okay, because eventually I'm going to divide those into 2 groups. So I want to replicate my DNA. That happens in the S phase, or the DNA synthesis phase. Okay. Now, what else do we have in the cell that needs to be replicated? Well, how about the organelles? So mitochondria, centrioles, vacuoles, all those need to be. And that happens in the G2 phase, where you replicate your organelles, because again, each cell needs the materials. Okay? So once you have your DNA replicated, once you have your organelles replicated, you can go ahead and go on and divide. So this portion here in the blue is the portion that we call mitosis. Okay, so we have the first phase is prophase. So here's up at the top. So I'm going to kind of move this and see if I can, hold on a second. Go back to my mouse quick. See if I can move this down. So this part of mitosis is when the chromosomes actually start to become visible. So one thing that I want to highlight here, actually I'll stamp it, would be, do you see the word chromatin? And do you see the word chromosome? Okay, so the word chromatin, remember, is when it's stringy. And that's the form it has to be in order to replicate it. So it has to be the double helix that we've learned about. But then when we want to divide it, so how do we get that plate of spaghetti so we can actually divide it? You need to have it in chromosomes, so it coils up into a chromosome. And so that's why you can actually see it under the microscope, because it looks all chunky. And so the chromosomes start to appear, your nuclear membrane disappears, and the centrioles, the cowboys, start to migrate towards the poles, because remember, they're going to form the spindles that pull those chromosomes apart. Metaphase, really easy. So again, instead of, remember, here's my dance. So instead of being together, now I'm going to line them up in the middle. And something that's really important, remember that you have double of everything. So of chromosome 1, you have 2 copies of chromosome 1. So you want to make sure that when those are lined up, that 1 copy goes to one side and 1 copy goes to the other side. So as far as how they line up, it's really important that you have 1 of each. Okay, so for example, with the kids, if I'm making sure everything's equal, they each get a big present, a small present, that sort of thing. So we call those, though, to keep in mind, we call those sister chromatids. Okay, sister chromatids mean sisters. Okay, and chromatid is just 1 chromosome. Okay, and they're lined up in the middle. That's metaphase. So when they start to get pulled apart, they actually form like almost in a V shape as they're being pulled. They're pulled in the middle, okay, by those spindle fibers, okay? Those spindle fibers that are extended from the centrioles are pulling those chromosomes apart. That's anaphase. Okay, that's anaphase, friends. And anaphase has that characteristic V shape in either direction. Now, something that happens, that can actually happen from anaphase all the way to the end here, is that the cell can actually start pinching in if you're an animal cell, okay? So in telophase, we have pretty much prophase backwards, okay? But in that time, you can actually get the division of the cytoplasm, okay? So you have mitosis is really the division of the nucleus, and cytokinesis is really the division of the cytoplasm. So you will start getting pinching in happening in an animal cell, but we don't pinch in on a plant cell. Anybody know why? Well, you can't when you're a cell wall. So do you see how it pinches in right here? Okay, but in a plant cell, they have something called cell plate formation. Okay, so let me go back. Anaphase, we've pulled them apart. Telophase, we're going to go backwards from prophase. We're going to be just chromosomes. We're going to reform the nuclear membrane. Okay, and it's going to go back from being chromosomes to chromatin. Okay, but we also then need to divide the 2 cells, which is the cytokinesis part right here. Okay, the cytokinesis. Now, a little bit lower on your page here, I'm moving, hold on, let me clear all my drawings. A little bit lower on the page is the cytoplasm division and the difference. So we call the pinching, and I think about it like a balloon, like pinching the middle of a balloon when someone's making a balloon animal. That's called furrowing in an animal cell. And an animal cell, again, can do that because it has a cell membrane. Plant cells can still have the pinching, but they have to build their cell wall from the inside out. So they actually lay the bricks on the inside out, and you can actually see that on a microscope. In fact, that virtual lab, you can actually see one of them, and you'll see this dark line in between. Okay, so this piece on the shortening of chromosomes, so this is kind of interesting. We'll talk about that a little bit more when we talk about cancer. I have included the answer key for this piece. If you like this organization maybe a little bit better, it's kind of backwards of what we did, but it might be a little bit better for you, and I'll include the answer key on this as well. Okay, hope that helps. See ya.